Good morning, wherever you are. This is Miles Carter from ConferenceHype. Our segment will focus on CD96, marks a phenotypically distinct checkpoint-associated HCV-specific CD8 plus T-cell subset featuring memory-associated states. From the 2026 edition of Frontiers in Immunology. Background, introduction, hepatitis C virus infection frequently progresses to chronicity, where persistent antigen exposure drives differentiation of virus-specific CD8 plus T cells toward an exhausted phenotype. Methods to evaluate the role of CD96 across infection stages. We used HCV-specific MHC class I tetramers combined with high-dimensional flow cytometry to phenotypically profile virus-specific CD8 plus T cells ex vivo in 32 patients spanning acute, subacute, chronic, spontaneously resolved infection, and post-treatment sustained virological response. Results: CD96 plus HCV-specific CD8 plus T cells consistently increased relative to bulk across all stages of HCV infection, with approximately fourfold higher frequencies than bulk CD96 plus CD8 plus T cells, and highest frequencies of 5,113% observed in the chronic group. Discussion: these findings identify CD96 as a marker of a phenotypically distinct checkpoint-associated HCV-specific CD8 plus T-cell subset, featuring memory-associated rather than terminally exhausted states. From the 2026 edition of Frontiers in Immunology. Background: early developmental stages of teleost fish represent vulnerable phases of the life cycle, characterized by exposure to fluctuating abiotic and biotic environmental conditions. Methods: in the present study, we analyzed the expression of 13 SNAD1 genes during embryonic and early larval development of common carp. Results: in contrast, SNAD409, SNAD208, and SNAD962 displayed delayed post-zygotic activation, with increased expression observed during later developmental stages coinciding with post-hatching maturation of immune-related physiological processes and establishment of host defense mechanisms. Discussion: the dynamic and stage-dependent expression of SNAD1 genes reveals distinct developmental regulation patterns and highlights substantial heterogeneity within the carp SNAD1 gene family during ontogeny. From the 2026 edition of Frontiers in Immunology. Background: autoimmune encephalitis is clinically heterogeneous, and its early diagnosis and prognostication remain challenging. Methods: this retrospective cohort study included patients who fulfilled the criteria for at least possible I according to the 2016 Grouse criteria and had their CSF IL-6 measured. Results: the cohort comprised 55 I cases. Discussion: CSF IL-6 is a useful marker for early eye diagnosis, and CSF IL-6-1M may serve as a practical biomarker for treatment response and prognosis, particularly for antibody-negative eye. From the 2026 edition of Frontiers in Immunology. Background: 1-carbon metabolism has emerged as a critical interface between tumor metabolic adaptation and anti-tumor immunity. Methods: beyond its canonical role in nucleotide biosynthesis and redox balance, this metabolic network regulates methyl donor availability, epigenetic programming, and immune cell state transitions within the tumor microenvironment. Results: recent studies show that tumor cells can outcompete T cells for methionine, thereby depleting intracellular S-adenosylmethionine, impairing histone methylation, and driving effector dysfunction or exhaustion. Discussion: in this review, we discuss how 1-carbon metabolism governs T-cell fitness, epigenetic regulation, and the broader tumor immune ecosystem, and we highlight emerging translational strategies for exploiting this pathway in cancer immunotherapy.
This is Conference Hype. Stay with us, coverage continues.
From the 2026 edition of Frontiers in Immunology. Background: immune checkpoint inhibitors have revolutionized the oncological landscape by disrupting inhibitory pathways, notably programmed cell death protein 1, programmed death ligand 1, and cytotoxic T lymphocyte-associated antigen 4 pathways, thereby reinvigorating host anti-tumor immunity. Methods: although these agents have emerged as frontline standard therapies for malignancies, their clinical utility remains limited. Results: interpatient therapeutic variability is inextricably linked to the composition and functional capacity of the gut microbiome. Discussion: such rigorous validation would enable the development of precise microbial interventions that maximize therapeutic gains while minimizing the incidence of adverse reactions. From the 2026 edition of Frontiers in Immunology. Background: the gut microbiota is a key regulator of human health and disease, exhibiting considerable functional diversity. Methods: high-fiber diets are known to profoundly reshape the gut microbial composition and its metabolic output, notably enhancing the production of short-chain fatty acids. Results: these SCFAs mediate communication along the gut-lung axis through multiple mechanisms, including epigenetic modifications, receptor-mediated signaling, and immunomodulation. Discussion: elucidating the mechanisms of microbial metabolites within the gut-lung axis through multiomics integration represents a promising option for developing targeted nutritional interventions, particularly for pediatric and treatment-resistant asthma. From the 2026 edition of Frontiers in Immunology. Background: rheumatoid arthritis is characterized by persistent synovial inflammation in which fibroblast-like synoviocytes and B cells interact within a potentially genotoxic microenvironment. Methods: this study investigates how naive B cells respond to cytotoxic stress when supported by RAW-FLS in a co-culture model, using viability assays, time-resolved gamma-H2AX MFI, RT-qPCR of DNA damage response and differentiation genes, and flow cytometric analysis of subsets and surface markers. Results: prolonged co-culture was associated with the expansion of B-cell-enriched compartments derived from naive B cells, and with increased expression of stromal markers CD90 and podoplanin on both B cells and RAW-FLS, while IL-10 secretion was reduced under cytotoxic conditions. Discussion: together, these findings define differential stress responses and phenotypic adaptations of B cells and RAW-FLS within an inflammatory co-culture system, and suggest that genotoxic stress may contribute to remodeling of immune stromal interactions in the rheumatoid synovium. From the 2026 edition of Frontiers in Immunology. Background: introduction: acetyl tributyl citrate is a widely used plasticizer ubiquitously present in the environment. Methods: we integrated network toxicology analysis, single-cell RNA sequencing, bulk RNA-seq data, and clinical datasets to explore the prognostic role of ATBC-related genes in testicular seminoma. Results: PDK1 and OARD1 were confirmed as ATBC-associated prognostic genes for testicular seminoma. Discussion: in conclusion, PDK1 and OARD1 are reliable prognostic biomarkers for testicular seminoma.
This is ConferenceHype. Stay with us, coverage continues.
ConferenceHype is interactive AI commentary only. It is not reporting, journalism, medical education, clinical guidance, scientific validation, legal advice, or financial advice. ConferenceHype is independent and is not affiliated with conference organizers, presenters, sponsors, or exhibitors. From the 2026 edition of Frontiers in Immunology. Background: retinol-binding protein 3 is a photoreceptor-derived glycolipoprotein that plays a central role in retinoid trafficking between photoreceptors and the retinal pigment epithelium. Methods: beyond its canonical role in the visual cycle, emerging evidence suggests that RBP3 may participate in retinal metabolic regulation, neurovascular protection, inflammatory modulation, and disease-related immune responses. Results: this review critically summarizes current evidence regarding RBP3 in diabetic retinopathy, retinal degeneration, pathological myopia, primary congenital glaucoma, and uveitis. Discussion: future studies should clarify disease stage-specific roles, receptor-mediated mechanisms, safety profiles, and clinical utility through large-scale prospective and mechanistic studies. From the 2026 edition of Frontiers in Immunology. Background: introduction: mushroom consumption has been associated with various health benefits due to their recognized nutritional value. Methods: male BALBC mice aged 4-5 weeks were divided into 5 groups: normal control, ciprofloxacin treated, natural recovery, ACP treatment, and ACP-derived fecal microbiota transplantation. Results: CIP administration markedly decreased bacterial diversity and richness, elevating pathogenic bacteria that persisted in the natural recovery group. Discussion: these findings highlight the prebiotic potential of ACP in restoring intestinal health, with therapeutic effects transferable via fecal microbiota transplantation, supporting their application as functional food ingredients for gut microbiota restoration. From the 2026 edition of Frontiers in Immunology. Background: with the widespread use of novel therapeutic agents, an increasing number of drugs have been confirmed to be associated with interstitial lung disease. Methods: we performed a signal mining study using data from the FDA Adverse Event Reporting System database from Q1 2004 to Q1 2025. Results: a total of 513 patients with ILD related to HNSCC treatment in the FAERS database were included. Discussion: ILD reports associated with HNSCC treatment were mainly concentrated among anti-EGFR monoclonal antibodies and PD-1/PD-L1 inhibitors, with the early treatment stage representing a critical window that warrants strengthened monitoring for ILD in clinical practice. From the 2026 edition of Frontiers in Immunology. Background: asthma remains a heterogeneous inflammatory airway disease, and current therapies, including biologics, benefit only selected patient subsets. Methods: we integrated cis-EQTL and cis-pQTL instruments for 585 druggable genes with asthma genome-wide association data from FinGen, EBI, and UK Biobank, comprising more than 1.1 million participants in total, using Mendelian randomization, colocalization, and SMR-HEIDI analyses. Results: CD83 emerged as the most consistently supported candidate across the genetic prioritization pipeline. Discussion: these findings prioritize CD83 in antigen-presenting cells as a genetically supported candidate therapeutic target in asthma. This is Conference Hype. Stay with us, coverage continues. That's it for now for Conference Hype's coverage of the January 2026 issue of Frontiers in Immunology. If anything was missed, tell us which article deserves follow-up. Tag us on X. Add your perspective in the comments, share this review with your clinical team, like the video, and subscribe with notifications turned on for the next journal broadcast.